these are some of the things that I believe we covered, or rather tried to explore and discuss throughout this, the ideas of sustainability, what makes an ecosystem. Specifically talking about the pig, we looked at which pig would decompose the fastest and what things would be related to that, the interconnectedness of different organisms and humans, and subsequently the ideas of succession. So starting off with the major part of this program, the pigs, 2 pigs. I'm just going to simplify it to Devon at this point, Devon and Porky. So these were our conditions, left out in the woods in a 3 foot by 5 foot box. All 4 walls were covered, a floor, and chicken wire was placed on top of each box. There were 2 bags of sand placed under each pig. They had been laid down on their backs with a slit. Everything was removed, I think, except the kidneys, and obviously bones and meat and flesh. And then a liver, heart, stomach, tongue, and a pint of blood were added inside the body cavity after each pigs were placed down. Additionally, Porky was covered with a layer of window screen, and we wanted to explore the effects of that, but subsequently removed that on July 20th to further investigate differences in succession. So, what would happen and why? And to do this and to explore how things would happen, we looked at several quantities, including the weight of the pig, the pH of the, it says soil, I meant sand, so the pH of the sand on certain days. We looked at the species and the number of them and types of species, weather conditions and rainfall, which is kind of related, the temperatures inside and directly outside the pig, with probes, which I'll explain later on, general appearances and infrared and heat information, both of which we have many photos of. So starting on day 1, we have each pig side by side. You can see Porky's covered in the screen, Devon is not. They each started out at about 52 pounds with pH of the high 8.5s. Quickly, we noticed that Devon was swarmed by flies and had many landing on him. Soon, Devon was completely covered in egg masses and then covered in maggots. And you can see that the heat signature picking up by the infrared camera is quite a lot more than Porky, because at this time Porky was wasn't really touched by anything. There was nothing that really got into it. There may have been a few flies, maybe a beetle here and there through some of the holes in the box, but there wasn't much. And these still had a high pH, closer to 9. And then again, Devon was quickly consumed while Porky was still left kind of untouched. At this point, we did see that Porky was experiencing some bloating, mostly due to, or we assume it's probably bacterial waste and discharge and things like that, which was causing the bloating and some of the foaming that we could see inside the body cavity. At this point, Devon was basically a pile of bones and was still very hot in certain parts because there were still several masses of maggots. And then Porky, as you can see, you might not be able to see it in the actual photos, but in the infrared screening, you can see all those dots are flies that are trying to get in. And we assume that they were a lot more at this point because Devon as a resource was being depleted and they needed another resource. So again, further continuation, Porky is very large at this point, lots and lots and lots of bloating, and some of the egg masses, we started to see some egg masses as some flies were able to get into the box and were able to kind of colonize that area. And this was, that was July 20th, that was after we took those photos, the screen was removed, and we quickly saw, as we do with Devon, flies quickly landed on Porky, kind of colonizing, laying egg masses, and within 24 hours became something to what we similarly saw to Devon. The temperature probes. So we had 2 temperature probes in and out of each pig. There was one in and one outside of each pig, so 4 temperature probes in total, and we used this to measure trends of temperature. So this was the internal temperature of Devon. The quick peak, you can see that was as both pigs were heating up. You'll see that also with Porky because they were both refrigerated. And you see incredible fluctuations because there were so many insects, so many bugs. Examples, there were road beetles, carrion beetles, blowflies, there were some hornets, bees. These all contributed to extra heat and extra produced energy as the pig was being consumed. Plus the probe was able to freely move, which could have caused these spikes and troughs. Porky was covered, as I said, until the 20th, and so we hypothesized that this kind of stability that we saw was due to the fact that there was nothing really there, and that any changes were mostly due to average day and night temperatures. But as you can see, at the 20th, or around the 20th, after that, a little bit towards the 21st, you can see a major spike, and then many fluctuations, as we saw with Devon, which we contributed, and that kind of furthered the hypothesis that these fluctuations were due to the organisms. And then both the external temperatures kind of fluctuated with day and night cycles. And that's Devon and Porky. Similar thing, there's just more data points because we had it out longer. The next major thing we did were the horseshoe crabs. So my uncle and Suzanne came and presented on these horseshoe crabs using the article Jurassic Beach as the focus of their presentation, as well as talking to Dr. Anthony Fredericks of York College. We explored many things about the biology, the evolutionary background, the importance, population changes, and geographic concentrations of horseshoe crabs. So the biology of horseshoe crabs, we explored the idea that they're arthropods and are more related to spiders rather than crabs and lobsters, do not contain hemoglobin and therefore contain hemocyanin, which is a copper-based blood which subsequently makes their blood a cyan or blue color. And the physiological features, that their eyes can see both visible light and light in the ultraviolet spectrum, as well as are photosensitive and can sense light intensity. And the horseshoe crabs have been around for hundreds of millions of years and have outlasted the dinosaurs as well as many other species, and have even outlasted several mass extinctions, one of which was being the dinosaurs. So the horseshoe crabs, we found out and learned, were extremely important, because they contain Limulus amebicide lysate, which is used in the sterilization of medical equipment, contact lenses, injections and immunizations, and many more. We learned that these creatures are basically incredibly useful and incredibly important, especially in the medical field, for human well-being and overall health. So horseshoe crabs concentrate, or at least the one of 4 species found worldwide that we're talking about, concentrate mostly in the Delaware Bay area of the eastern U.S. They have had points of concern due to diminishing populations, and there have been many local organizations and citizens which help the horseshoe crabs as they usually get washed up on the beach during their mating seasons, which has ended around now. The amount out on the beaches are very limited at this point, and there have been several laws regarding over-farming and the diminishing populations because the use of the Limulus amebicide lysate is federally regulated and must be used in the sterilization of medical equipment. And this is just a quick tidbit. Dr. Anthony Fredericks came and answered some of our questions and provided insight on some of the horseshoe crab facts and some of the things that we weren't that we didn't necessarily understand. This was something we also did. We went to the Stony Brook Millstone Watershed Association. Jeff here kind of talked us through the conservation efforts of the watershed, and then we went on to test clean and test the ways to test for clean and healthy water through biotic and abiotic factors. So the watershed has a LEED Platinum rating, which means that it's very eco-friendly. Many of the things include proper placement for recycled products. They use rainwater for the bathrooms, or rather the toilets. The sink water is heated by pipes that run outside the building and are heated by the sun. And they use things like roof gardens and solar panels to maximize rainwater and sunlight energy. So the stream analysis, by testing the organisms in the water, we were able to tell how healthy of an ecosystem it is. And some of the insects and animals that we found, crawfish, leeches, wolf spiders, minnows, and several more of these smaller insects that were very susceptible to pollution changes. So it was very good that we found some of these smaller, more susceptible individuals. And then the abiotic factors, using certain instruments, we were able to test salinity, which had an average of about 5 to 6 parts per thousand, turbidity, nitrate and phosphate levels, temperature, and a pH of the stream, which we all found in both groups that was normal and indicated a healthy and proper ecosystem for the aquatic organisms that were made to live there. And these are just a sample of the tools we used: salinity tester, thermometers, and a pH testing kit. And the last thing that we kind of did was talk with Dr. Druckenbrod, who talked about thermology and infrared radiation, climate change, and the greenhouse effect. So thermology, not only does visible light penetrate the atmosphere, frequencies below that of visible light, known as infrared radiation, also penetrate, and most of this radiation is absorbed by lakes and the ground, and us, when we go outside and we feel hot, that's us absorbing the infrared radiation from the sun. But some of it is reflected back, and it is also absorbed by clouds and other animals that are flying in the sky. And so this is what causes higher altitudes to become cooler, because there's not as much infrared radiation. And the greenhouse effect and the components of the atmosphere we talked about: nitrogen, which is 78% of the atmosphere, oxygen 21%, argon, carbon dioxide, and helium, so on. These are several major gases that are part of the atmosphere, and there are many more which we call trace gases, because their percentages are not high enough for us to really, not necessarily care, but they're so small. And the greenhouse gases include carbon dioxide, methane, and water vapor, which, due to their nature, are very good at trapping heat and trapping this infrared radiation. And as more is released, more radiation is trapped, and therefore the average temperature of Earth rises and continues to rise. And this all ties together to global warming, which the greenhouse gases can quickly devastate the colder climates and environments. As you can see, this polar bear, who doesn't necessarily have a viable way of traversing. And so with all of this, the takeaway, I guess we can talk about, is that organisms, many organisms, and not just one singular organism, is needed for the suspension of an ecosystem. Ecosystems are constantly changing, and even little factors such as a couple hundred parts per million of carbon dioxide can change the temperature and change the weather patterns drastically. There are many organisms like the horseshoe crab that have a huge impact on human health and well-being, and are should be looked at as something that is great, and not something that we shouldn't care about, something that we need to protect and conserve. And we can see clearly with the pigs that nothing is wasted. Ecosystems are very good at turning everything, like the dead pig, into resources that they can use. And overall, with the idea of sustainability, environmental impacts deeply strain how well and how effective different organisms can grow and survive as species. And that's just bibliography.
Questions?
Thank you.
Questions for Alan?
Okay.
Guess you were all there, right?